So I welcome you to today's lesson, and I hope we are going to learn things by the end of this tutorial or lesson.
Access recorded videos.
Sorry?
Where do we access the recorded videos?
They are there on campus classes.
All right.
Okay. So welcome once more.
Thank you.
And for today, we are looking at gene expression. If you can recall from the previous lessons, you do understand that gene expression happens via two processes, and that's translation and transcription. So gene expression happens or occurs via transcription and translation. And so specifically for today, what we are looking at is how gene expression is regulated. How gene expression is regulated. So in short, we are looking at the regulation of gene expression. And so it would serve good if we can recall the process of gene expression. We can recall the process of gene expression. Okay, so let's avoid scribbling on the screen. Okay, so I said gene expression happens via transcription and translation. And this together with DNA replication could be summed up in what is known as the central dogma of molecular biology. And when we looked at translation, we did define what a gene is. And a gene is simply a segment or a part of DNA that codes for a complete polypeptide. A gene is a part or segment of DNA which codes for a protein. That's a gene. And when we look at gene expression, hence, it's simply the production of proteins, because ultimately from replication to transcription to translation, what we are going to have ultimately is the production of proteins. And the proteins are actually the active cell machinery. They are the active cell machinery. So gene expression is simply the production of proteins. So for example, we find enzymes are being produced. So simply gene expression, production of proteins using the genetic code. That's gene expression. And then we have gene regulation. Gene regulation. This simply is the mechanism by which cells use to switch the processes of transcription and translation, either on or off. So gene regulation simply refers to the mechanism that cells use to switch the process of transcription and translation on and off. Okay, so why is gene regulation important? Anyone who knows why gene regulation is important? Guys, anyone? Why should the expression be regulated? Yes, you can go ahead. I can see a hand.
To avoid the waste of protein.
Okay, thank you. So
Uh,
that's true. Yes, go ahead.
I think gene regulation is important as it allows an organism to just express certain proteins only when they are needed.
Okay, thank you very much. There was another hand. You can go ahead.
Okay, gene expression is important because it allows specific proteins to be used at the specific time, thereby preventing waste of materials and energy.
Okay, thank you very much. So you are all correct. You are all correct. Thank you. So that's gene regulation. It is very important. As you have already mentioned, it allows an individual to be able to produce certain proteins only when they are needed. So only when they are needed, they can be produced. So when you look at gene expression regulation, we find that in prokaryotes, what happens is that gene regulation is primarily takes place or regulated at the transcriptional level. However, in eukaryotes, it is regulated at many levels. It is regulated at many levels. However, for our course, we are going to concentrate on regulation, gene regulation in prokaryotes. And we are going to use the bacteria. We're going to use E. coli as an example. We're going to use E. coli as an example. So gene regulation involves the cellular processes which control the rate and the manner at which gene expression happens. So it's a complex set of interactions. Yes, there's a hand. You can go ahead. Mwanga, go ahead. Okay, so gene regulation of gene expression is a complex process involving a set of interactions. These interactions will include the genes, the RNA molecules, the proteins, and other components that are in the expression system. And hence, this is going to lead to a determination of when and where these specific genes are activated, and also the amount of protein that is needed to be produced. And hence, we find that the significance of gene regulation is that it helps in avoiding wastage of materials and energy as well. So the regulation of gene expression helps to avoid wastage of materials and energy. In other words, we are saying gene regulation conserves energy and as well as space and materials. So as we said earlier, due to gene expression, some of the products may not be produced at certain times or by certain types of cells because they are not needed by those cells. And then we find that certain gene products are only needed in certain cellular conditions. Hence, they are just going to be produced in those conditions. When those cellular conditions are not there, the gene product won't be produced. And hence, we are conserving energy, we are conserving space, and we are conserving materials. So the mechanisms of gene regulation include regulating the rate at which transcription happens, regulating the RNA processing process, and also regulating the stability of messenger RNA, and also regulating the rate of translation. Remember the process of transcription. And we said that in prokaryotes, regulation is primarily at the transcriptional level. And so during that process of transcription, the gene can be regulated. And then also during the post-transcriptional modifications or RNA processing, regulation can also happen. And also the stability of messenger RNA. Remember, the post-transcriptional modifications add to the stability of messenger RNA. We know that after messenger RNA has done its job, after the process of translation has happened, messenger RNA need to be degraded. So we have the stability of messenger RNA playing a role. So the mechanisms of gene regulation includes regulating the stability of messenger RNA. And then it also includes regulating the rate of translation, So that's what is being depicted on the top right diagram there. So you have transcriptional control, you have RNA processing control, you have RNA transportation control, and translational control as well. So what are the components of components that are found in an Operon? So the classical example that we give of gene regulation system is the lactose operon. The lactose operon, or simply Lac Operon. Lac Operon. And this is present in E. coli. This is present in E. coli. When we look at an Operon, it's simply a group of genes. So an Operon is simply a group of genes that are working together in order to carry out a series of related biochemical reactions. So an Operon is a group of genes, and these genes work together in order to carry out or conduct a series of reactions. That's an Operon. So when we talk about a Lac Operon, which is a lactose operon, this simply means it's a group of lactose metabolizing a group of structural genes. And those structural genes are controlled by a common regulator gene. The structural genes in a Lac Operon are regulated by a common regulator gene. So as mentioned earlier, gene expression is regulated primarily at the transcriptional level in prokaryotes. And the process by which these genes are activated or deactivated is well characterized in E. coli. So bacteria have three types of genes. And these are the structural genes that we're going to concentrate on. Now, these structural genes code for the synthesis of specific proteins. Are going to code for specific proteins, for the synthesis of specific proteins. They are also operator genes. So bacteria have types of genes: structural genes, operator genes. These are genes that bound by the repressor. They're bound by the repressor. And then we have the regulator genes. These code for the repressor protein. So in prokaryotes, regulation is achieved by means of genetic units, and these are what we call operons, and alongside a number of related mechanisms. And I've explained what an operon is. Simply a group of genes that work together to carry out a series of reactions. And these reactions are related, so related biochemical reactions. So as you can see in the diagram there, you are going to realize that an operon will consist of a promoter, it's going to consist of an operator, and it is also going to consist of a regulator and also structural genes. So an operon consists of a promoter, consists of an operator, consists of a regulator, and also consists of structural genes, the structural genes. And these structural genes are going to be transcribed into messenger RNA, into messenger RNA. And then from transcription, we know during the process of translation, there's going to be the formation of polypeptide chains of amino acids, leading to the production of proteins. So these proteins, as depicted there, there can be various types of proteins that produce these structural genes. So that's what an operon consists of. So the other components that we're going to find in an operon include what is known as a catabolite activator protein, CAP, abbreviated as CAP. So catabolite activator protein. So the CAP binds in the presence of a CAMP, cyclic AMP, cyclic adenosine monophosphate. So the CAP binds in the presence of cyclic AMP to sequence close to the promoter. So remember, you have the promoter there. So it's going to bind to the sequence close to the promoter. And its function is simply to enhance the ability of RNA enzyme to bind to the promoter. It enhances the ability of RNA to bind to a promoter. So that's a CAMP. So catabolite activator protein. It's also known as a camp response protein, or CRP. So it binds CAMP. So in short, we're saying CAP is an allosteric protein with CAMP as an effector. Catabolite activator protein is an allosteric protein with CAMP as an effector. So in short, we're saying the CAMP alone does not bind to DNA, but binding of the effector CAMP to CAP changes its conformation such that it can then bind to DNA. Remember, we're saying it's an allosteric protein with a cyclic AMP. So what happens is that the CAMP binds to the effector, or the binding of the effector CAMP to CAP changes its conformation such that it is then able to bind to DNA. And we are going to see that the level of CAMP in cells is reduced in the presence of glucose. In the presence of glucose, CAMP is reduced such that there is no stimulation of transcription from CAMP responsive operons takes place. So we have CAMP as one of the components. The other components is a promoter, which is a sequence on DNA template strand that serves as an attachment point for RNA polymerase. So RNA polymerase is an enzyme. So it's an attachment point. The promoter is an attachment point for RNA polymerase. Okay. And then we have the replacer. The repressor. So this is a DNA or RNA binding protein which binds to the operator. When it binds to the operator, inhibiting expression of the genes, inhibiting the expression of the genes. So we see that the initiation of transcription of the Lac Operon is actually controlled by the Lac repressor. So the repressor is going to bind to the operator. Now this operator, as we can see, is just adjacent to the promoter, just adjacent to the promoter. So when this Lac repressor binds to the operator, this prevents the RNA polymerase. Remember, we have the RNA polymerase here. This prevents the RNA polymerase from binding to the promoter. It's going to prevent RNA polymerase binding to the promoter. And we see that this DNA binding is actually sensitive to the presence of lactose. Now that's called Lac Operon. So the repressor binds DNA in the absence of lactose, but not in the presence of lactose. So that's the repressor. And then we have what is known as the co-repressor, which is simply the molecule which binds and activates the repressor. So co-repressor simply binds and activates the repressor that we've talked above there. And then we have what is known as the inducer. We have the inducer. So the inducer is a molecule which binds and inactivates the repressor. The inducer binds to the repressor and hence is able to inactivate the repressor. So as given there, we have the repressor which cannot bind to the operator by itself. So it requires this co-repressor. And when that repressor, when the co-repressor and the repressor form a complex, that complex binds to the operator and replaces enzyme synthesis. So we have what is known as negative control, and that negative control is actually mediated by the repressors. The negative control is mediated by repressors. And then we have positive control, which is mediated by another class of regulatory or allosteric proteins, which are known as the activators, that can bind to DNA and stimulate the initiation of transcription. And these enhance the binding of RNA polymerase to the promoter and hence increase the level of transcriptional initiation. They increase the level of transcriptional initiation. So this is a Lac Operon, which uses glucose as a preference whenever it is available. The enzymes for glucose utilization are made constitutively. So we find that in the absence of glucose, E. coli is able to produce enzymes and other proteins in order that they can utilize other carbohydrates, for example, lactose. The Lac Operon contains genes which code for enzymes and proteins that are involved in the metabolism of lactose and its transportation as well. So there's three structural genes, as we can see there in the bottom left. There's three structural genes. We have LAC-Z. The LAC-Z codes for beta-galactosidase enzyme, which is a lactase. Now this beta galactosidase enzyme breaks down lactose into glucose and galactose. Remember, lactose is a disaccharide formed from beta galactose, formed from beta galactose and either beta glucose or alpha glucose. So it's a disaccharide of galactose and glucose. So the beta galactosidase enzyme hydrolyzes or breaks down lactose into glucose and galactose. So we have the LAC-Z gene, which codes for beta galactosidase enzyme. And then we have LAC-Y. The LAC-Y codes for lactose permease, the enzyme lactose permease. That's LAC-Y. So LAC-Y gene will encode this lactose permease, which is simply a lactose transporter. It is a membrane-bound transport protein, also known as a beta galactoside permease. So this permease pumps lactose into the E. coli cell. And then we have the LAC-A gene. This codes for transacetylase, beta galactoside transacetylase. So this modifies the lactose. It modifies the lactose when it is inside the E. coli cell. So it adds acetyl group to the galactose, and that's known as a beta galactoside transacetylase. So this one modifies the lactose. So those are the structural genes. So we see that the Lac Operon works with the promoter sequence. As we can see, it works with the promoter sequence. It works with an operator sequence. And also, in addition to that, it also works with the terminator sequence. So this is the Lac Operon and its components. So we've laid a foundation of the Lac Operon. And in our preamble in the foundation is cardinal to the understanding of everything that we are going to look at further. So as mentioned earlier, we see that E. coli prefers glucose as a source of energy. So in the absence of glucose, the cell is going to use lactose instead as a source of energy. So now, for the cell to be able to use the lactose, for the cell to be able to use that lactose, it must first produce the enzymes which are needed for the mobilization of the lactose and also for the digestion of the lactose. So before the cell uses the lactose, it has to produce the enzymes that are needed for mobilization of lactose and for the hydrolysis or digestion of the lactose. So additionally, and looking at what we've looked at so far, the Lac Operon has to ensure that the cell uses energy needed to utilize that lactose only when it is necessary for it to do so. So remember, the regulation has to conserve. It conserves energy, it conserves space, it conserves material so that no material is lost. Therefore, if that is the case, it means that in the presence of glucose, the Lac Operon is switched off. In the presence of glucose, the Lac Operon is switched off. In the absence of glucose and in the presence of galactose, the Lac Operon is switched on. So there are two regulators activate or deactivate the operon in response to lactose and glucose levels. And we've explained the Lac repressor, we've explained CMP, CAP, we've explained these. So the lac repressor senses the levels of lactose indirectly through its isomer. And this isomer is known as allolactose. It is known as allolactose. So allolactose will act as an inducer in activating the lac repressor once bound to it. So the Lac Operon senses lactose levels through allolactose. And this allolactose acts as an inducer, leading to the inactivation of the lac repressor, leading to an inactivated replacer. So this is what is being shown there. The allolactose binding to the repressor. The allolactose binding to the replacer will inactivate the replacer. So you have the inactivated replacer there. And then we have the CAP, which we have explained. The CAP senses glucose levels indirectly, as we have explained, through the molecule cyclic AMP. Remember, we mentioned that the cyclic AMP will bind to the CAP. So the CAP binds close to the promoter and enhances RNA polymerase binding. So the role of CAP is that it enhances the RNA polymerase binding. In short, we are saying that in the presence of CAP, we expect the transcriptional level to be high. We expect the activity of RNA polymerase to be high as well. So that's what is being depicted in these two diagrams here. So it's, remember, we said the CAMP binding. When CAP binds, transcription need to proceed, or rather it enhances the activity of RNA polymerase. So what happens to the transcriptional process in the presence of glucose? So remember we mentioned that in the presence of glucose, the Lac Operon will be switched off. In the presence of glucose, the Lac Operon will be switched off. So we have the operator, we have the replacer binding. And hence when it binds there, remember this is in the presence of glucose, and the Lac Operon need to conserve material. The Lac Operon need to conserve space